Studies suggest blood thinner heparin can neutralize coronavirus. Researchers from the Rensselaer Polytechnic Institute in New York have found that a common drug already approved by the U.S. Food and Drug Administration may neutralize the novel coronavirus. The research, published on July 10 in the monthly peer-reviewed medical journal Antiviral Research, suggests that the blood thinner heparin can bind to the surface spike protein of the virus. SARS-CoV-2 uses this spike protein to bind to human cells, but heparin can reportedly bind to the spike protein and block infection from occurring. The drug can be used by patients as a nasal spray or through nebulizer, effectively lowering the odds of COVID-19 infection. According to Science Daily, heparin acts as a decoy, the way other treatments have been found to act as a decoy for viruses such as dengue, Zika, and influenza A. Robert Lindhart, the study's lead author and a professor of chemistry and chemical biology at the institute, said heparin could work as an early intervention to reduce the infection. The drug could be administered to coronavirus-positive patients who have yet to experience symptoms. However, the researchers also see this as a part of a larger antiviral strategy. Though there are 150 vaccine development efforts worldwide, Lindhart said there are many ways to combat a virus. With the right combination of treatments, including heparin, doctors may be able to control the disease until a COVID-19 vaccine is made widely available. Viruses infect cells by binding to a target on a cell surface, penetrating the membrane, then hijacking the cell from the inside in order to replicate the virus. Scientists have found that viruses may also end up binding to decoy molecules, as long as they are similar to the target cells. Decoy molecules neutralize the virus, rendering it unable to infect other cells or leave the decoy. The novel coronavirus is known to target the ACE2 receptor, and the newly published study shows that heparin can bind to the SARS-CoV-2 spike protein at 73 picomoles. Jonathan Dordick, a chemical and biological engineering professor at the institute, said the interaction between the spike protein and heparin is hundreds of thousands of times tighter than a typical antibody antigen. He explained, once it binds, it's not going to come off. The researchers have begun to test its antiviral activity and cytotoxicity in mammalian cells.